Hi guys, this lecture is about how to do different types of Punnett squares. We're going to do a monohybrid Punnett square, so a 2 by 2 square, and a dihybrid Punnett square, so a 4 by 4 square. But before we get there, I want to kind of go over where do we get the information to kind of predict what the offspring of those parental generations are going to be like. It basically goes back to our knowledge of meiosis, but we get some information, we inherit some information through chromosomes from our mom and from our dad. So when gametes are formed, we get 23 chromosomes in a gamete from mom and 23 from dad, and when they come together, fuse to form a zygote, we get a full 46 chromosomes, and those chromosomes share information about how we're going to develop based on how our parents look or how they developed. So to represent that and to kind of show that, we're going to just put mom up here and we'll put dad over here. Okay, so let's say that these are chromosomes, single chromosomes, that we get one from mom, one from dad in this instance. Let's just say that. So remember, a chromosome is really just a strand of DNA, and on that DNA we can actually break up the DNA into segments, and each of those segments are called genes. And let's say that right here we have a gene that determines, I don't know, hair color, and let's say that dad has brown hair, and let's say mom's a blonde. Okay, now when we put these what are called alleles together. Alleles are basically letters of our alphabet that we use to represent what is actually being expressed, or the information that we get from either mom or from dad. So essentially what we're looking at are actually alleles from those genes. And mom's going to give up an allele and dad's going to give up an allele, for instance, for hair color. All right. So let's just say, in this instance, that in order for dad to have had brown hair, he has to show a dominant allele. And we write dominant alleles in uppercase letters. So let's say that he had to have, we'll use B's in this case. He had to have a dominant, or an uppercase B, a dominant allele, in order to get brown hair. Well, that means that if he had to have at least one dominant allele to get brown hair color, then mom was out of luck. She had to have, and we're going to use the same letter, it's just uppercase or lowercase. Mom had to have no uppercase alleles, or no dominant alleles. So in order for her to have blonde, we'll say blonde is recessive, she had to have 2 lowercase alleles. Alright, so just to go over some terminology, we've got a couple of these words down here for you. Homozygous means the same. Homo in Greek and Latin means the same. And what's being the same? Well, this is a good example of a homozygous set of alleles because they are the same. And the way that we represent them would be 2 lowercase letters. So how many different combinations could you have of homozygous alleles? Just 2. We could have homozygous dominant. So in the case of the brown hair, we could have 2 uppercase or dominant alleles. And we don't know if dad has that or not, okay, but they would kind of look like this. So we could have a homozygous dominant individual that would have 2 uppercase letters, or 2 uppercase letters representing 2 alleles, or we could have homozygous recessive, which is what the mom is, and that would look like this if we're just using the alleles that, or the letters that we're using for these. Now we could have an individual that is heterozygous, and that means different, okay? And if you have an individual that is heterozygous, if we're using these letters to represent this allele, or these alleles, their alleles would look like this: big B, little b. See how they're different? Now the big thing that people tend to get hung up on is that we use the same letter just in different cases. A lot of people like to use different letters to represent the different alleles, but you need to stick with the same letters for each gene. If we're dealing with different genes, that's where it's okay to use different letters, 1 letter for each gene. But you still need to use that uppercase and lowercase letter combination to show which is the dominant allele being expressed and which is the recessive allele, which would be masked by the dominant allele. Okay. The only trick to that is, however, the only exception to that is if there are two recessive alleles or homozygous recessive alleles, in which case they express themselves. Okay, so let's go back up here and figure out if mom and dad had a kid, what their kid would look like. What kind of hair color would their kid have? So let's just say that this represents the possibilities for the child's hair color. Well, when forming gametes, mom can really only give up the same kind of letter here. She's only got a little b or a recessive allele to give up. She doesn't have the large dominant allele that dad has. So either way, when gametes are forming, mom's going to give up a little b no matter what. That's all she's got to give. Okay. Dad, however, could be heterozygous or could be homozygous recessive. So his is going to be where the difference comes. Okay. And notice how I put this little b second. Whenever we write these combinations of alleles, we always write capital or dominance first over recessive. So the more dominant alleles are going to always be written first next to the recessive alleles. So dad could give up one of these, okay, or, and let's say mom, here's another combination that could happen. Mom, remember, can only give up one of those, or maybe dad gives up this one. We don't know. Okay, and again, same thing here. Okay, dad could give up this one. Okay, and again, mom can only give up the smaller one. So the child could have either brown hair or could have blonde hair. We don't know. Okay, but that's the combination of alleles that could be given up by both parents. And remember, they can only give up one. And remember, going back to meiosis, when the gametes do fuse to form a zygote, mom gives up one allele randomly and dad will give up one allele randomly. And this is through independent assortment, when the chromosomes independently assort to the different poles and get pulled aside. So independent assortment definitely plays a big role right now in helping to give us that variance in our population and what we look like. One other thing before we go on, I'd like to talk a little bit about the difference between a character or characteristic and a trait. So let me just pull this over. Okay. A character is something that's much more generalized speaking. It's a general term that we use to describe the type of information that the allele is holding, but it's a generalized term. So for instance, maybe we're talking about hair color. Okay, that maybe that gene or that allele is going to determine hair color, but we're not specifically saying what color the hair is. We're just saying that that's what that gene is in control of. It could be skin color, it could be height, again, anything like that. A trait, however, is specific. It's actually going to tell you what eye color that child might have or that offspring might have, what hair color that offspring might have. So for instance, maybe that child or that offspring is going to have brown hair. So see how we're being very specific in naming the actual color that's going to be represented? A couple of other terms that we need to go over is genotype and phenotype. Genotype is actually talking about the specific alleles, what they're actually being described as. So for instance, if we go back to our hair color for our parents, we were using big B's, okay, and little b's, and there's different combinations, right? We have homozygous dominant, we can also have homozygous recessive, and then this would be heterozygous. So when we talk about the genotype, we're actually talking about, okay, what are the combinations of alleles specifically? When we talk about phenotype, however, we're actually talking about what is that individual going to look like? What does this mean, right? So is this person going to have brown hair, or is this person going to have blonde hair? Okay, so I'm going to erase this for now because we're going to actually use this for our Punnett square because we've basically gotten to that part now. Also, a monohybrid, that's what we're going to do now. Mono meaning one. Okay. What are we talking about when we say monohybrid? When we do a monohybrid cross, we're going to be looking at one specific gene or one characteristic. And in this case, I'm going to use the example of flower color. Okay. So let's say that our parents, are are going to be true breeding. So we're going to do a true breeding cross. So what does that mean? Well, if we do a true breeding cross, that means that the offspring of the first or even second generation that we find are going to be identical to their parents. So usually that means we're going to be seeing a lot of homozygous, either dominant or recessive alleles and phenotypes coming up. So they have to look exactly like their parents 100% of the time. So the only way that you're going to really be able to do that is if we have, say, let's say dominant P or uppercase P stands for purple. And we'll put a little p for, say, white flowers. So in order to get white, though, we have to have a homozygous recessive set of alleles. So I'm going to put 2 little p's there just to remind you of that. So let's say that our parents are true breeding. So I'm going to give you an example of that. When we do this, let's do dominant first. So let's say our mother or our female flower has 2, or is a homozygous dominant plant. We're going to cross that with a male part of the plant that's also homozygous dominant. So when we do this, it really doesn't matter if we put mom up here, dad up here, or dad down here, or whatever. When you get really deep into genetics, though, sometimes you do have to keep track of which one's the male, which one's the female for sex-linked genes and traits and things like that. But right now, we're just going to hit the basics. So we're going to put a P up here and a big P up here, and we can say that that's mom, and we're going to put a big P here and a big P there, all right? And basically what you're doing is you're going to bring a letter down if it's on top and over if it's on the side, okay? And again, we always write our most dominant allele first, but in this case, they're all big P's, okay? So genotypically, the genotype of this Punnett square, or this F1 generation, which is all of these guys, those are our offspring, the genotype is going to be big P, big P. Another way to say that is homozygous dominant. Our phenotype, they're all going to have purple flowers. Oops. Okay, so this is what we consider a true breeding plant, because even if we crossed any of these offspring with each other, we're still going to get a true breeding purple plant. And that's actually what Mendel did with his pea plants. So let's just erase this so I can give you another example. Now, this would be the same thing if the parents were homozygous recessive as well, okay? So let's do another cross. Let's say that we aren't true breeding. Let's say that our parents, or the parental flower, the parental generation flowers, are heterozygous. So what's that going to look like? Well, basically, one parent is going to look a lot like this. We got a big P and a little p. We're going to cross it with another plant that is also heterozygous for this. Genotypically, they're going to be heterozygous, but phenotypically they're going to have purple flowers. So they're going to look like each other, but genotypically they're going to be a little bit different. Or be able to produce different offspring. So we're going to do the same thing. We're going to put a big P there and a little p there, and that would take care of one of the parents. We'll put the same thing over here because that's exactly what the other parent is, and we're just going to bring them down and over like we did before. These are both dominant, so we should have 2 dominants there, and you can continue to fill this out if you wish. And down here we've got 2 recessive. So here's some more questions for you. What is this plant actually going to look like? Here is our F1 generation. We've got a homozygous dominant plant here. We've also got a homozygous recessive plant here. And we've got two possible, or 50% of the offspring that could be produced would be heterozygous. That's one thing that I forgot to tell you, is that each one of these boxes represents 25% of the overall possible amount of offsprings that could be expressed with that specific allele combination. So what do I mean by that? Basically, if I asked you, what is the probability that these 2 plants, if we cross them, would have, say, purple flowers? Okay. The offspring, what is the probability that the offspring would have purple flowers? Well, we have 25% here. Here's another one that could have purple flowers, so that's another 25%. And here's another 25% out of the whole, which would be 100%. So if we add those together, we have 75%, there's a 75% probability that the F1 generation of this parental cross would have purple flowers, and only 25% of that offspring would have white flowers. Phenotypically, so that's what that means phenotypically. What about genotypically? What's the probability that some of those offspring would be homozygous recessive? 25%. What's the probability that some of those offspring would be heterozygous? 50%, because remember, this represents 25 and this represents 25, so you got to add them together, okay, and so on. So let's take a look at what this means in terms of some other questions. What are the genotypes that we see here? Well, we've got homozygous dominant plants, we've also got some heterozygous plants, and we've got some homozygous recessive plants, okay? What are the phenotypes? Well, we've got 3/4 of the offspring, or 75%, are going to be purple flowers, and 1/4 of the offspring, or 25%, are going to be white flowers. Okay, so we can also demonstrate this information by looking at or asking what is the phenotypic ratio of this offspring set, and what is the genotypic ratio of this filial 1 or F1 offspring. So what does that mean? Well, when we write a ratio, we always use colons. Okay, so, and we always start with the most dominant set of alleles and work our way down to the most recessive set of alleles. So when we write our phenotypic ratio, we're asking how many of this offspring, how many different combinations of traits does this offspring set show? Well, that's pretty simple. We have 3 boxes here that show purple flowers. So, and since that's our most dominant set of alleles and our most dominant of the 2 possible traits that could be shown, we're going to write 3 because we have 3 individuals out of that whole group that would show a dominant trait. And then we have just the 1 individual that would show a recessive trait. Okay, or be white flowers. And remember, we're talking about, when we talk about phenotype, we're talking about what is expressed. What are we actually going to see? Okay, so we have a 3-to-1 ratio for phenotype. Now, what about genotypic ratio? Okay, well, genotype, remember, we're actually going to look at the different combinations of alleles that we've represented by through letters. So how many different types or combinations do we have? Well, we've got 1 here. We've got 1 here that's different, and these 2 are the same, okay? So we have basically, starting from our most dominant, okay, so this would be our most dominant, we have a 1, and that's going to represent this one, because that's our most dominant. The next most dominant are going to be actually these 2, and we have 2 boxes, or 2 individuals that represent that, so 2 there. And finally, our least dominant, or our most recessive, we only have 1 box there, or 1 individual that's going to represent that all those offspring that could have a homozygous recessive trait, okay? So we would write that like that. If you were just to see this set of numbers, a 1-to-2-to-1 ratio, you should know that the most dominant is this one, okay? And don't think of it as individuals per se, but if you wanted to think like that, just think that 25% is going to be is going to show a homozygous dominant in this case set of alleles, and this one is going to show a heterozygous. So 50% are going to be heterozygous in this cross. And finally, that last one would be 25%, would show a homozygous recessive set of alleles. Okay, so that's our basic monohybrid Punnett square. Let's go on and let's check out how to do a dihybrid Punnett square. And what I'm going to do, I'm just going to move this over. So what are we actually looking for when we do a dihybrid cross? Okay, so this is going to be a dihybrid cross. Okay, di means 2. So what are we actually looking at? Just like monohybrid, we're going to be looking at not 1, but now 2 different genes. Okay, so 2 different traits. Okay, and in this example, I'd like to look at, since we're on the subject of plants, I'd like to look at seed color and seed texture. Okay, so let's say that our seeds can either be green or yellow, and that green is dominant. Okay, so green, if we have green seeds, that means they're dominant. We're just going to use a big capitalized G to represent the dominant allele for green. Okay, and remember, if you're going to be green, then you either have to have two big G's or be homozygous dominant for this color for seed, or heterozygous, and we'll just write our little g there, in order to be green. And finally, yellow. Well, yellow would be recessive, so we would have two little g's. Okay. Now let's look at seed texture. Let's say that we have smooth, and let's say that's represented by a big T, and we have wrinkly, and that's going to be represented by two little t's, or a little t. Okay, not to confuse you, but this is what you would need in order to show this phenotype. Okay, and this is what you would need to show this phenotype. Okay, so this is going to look really complicated, but it isn't. I'm going to show you a trick to figure out how to actually set up your Punnett square. Okay, so let's say we match or we combine or cross two different individuals that are heterozygous for both of these traits. Okay, so they're going to have green, because that's the dominant, and smooth types of seeds. Okay, so they're going to have, and they're heterozygous, so that's what they're going to look like. So each individual is going to have a large G and a little g, okay, to represent its heterozygous green seed color. And finally, a big T and a little t to represent its smooth seed texture. Now, we're going to cross that with another heterozygous individual. So they're going to have the same genotype. Okay. Now, how are we going to set up our Punnett square? Okay, because this would not work if we were trying to put this and cram it into every single box. That's not going to work. So we're going to just use 1, since they both are the same, they have the same genotype. We're just going to use 1. We're just going to use one set of alleles to represent both. Okay, so this is kind of the trick. So I'm going to write this over here. So we have G, G, big T, little t. So here's the trick. You're going to do a combination where you start with the big G and you're going to combine it with the big T. That's our first allele combination. And when you write these, it's okay to write it exactly how you see it. So if the recessive allele comes first, then write it that way. It all straightens out in the end. I'll show you how. So big G to little t, that's our first one. Okay, then you go to the next allele. Little g to big T. Okay, so now, all right, so let me back up here. That's not what you want to do. So I'm going to cross that off. You always want to start, and that's usually the problem with a lot of people when they first start doing this, they always want to go to the next one. You got to do one allele first. I thought I'd show you first before you mess it up. So big G first goes to big T. Okay, we got that. So we need to finish up the big G though. Okay, so big G is going to pair up with the little t. Okay. Oops. So big G, little t. All right. So now our G is taken care of, our big G. Now we got to go to our little g. Well, little g is going to go and pair with big T, just like we did before, but now this is the right time to do it. So little g, big T. Same thing, it's going to pair up now with the next allele. Little g, little t. This right here is the combination of alleles that we're going to use for the boxes to set up our Punnett square with. So let's do that. All right, so let's plug these in. Okay, so we've got big G, big G, big T, right? Big G, little t. Then we have little g, big T, and then little g, little t, okay? So we're always going to put our two combination of alleles in each of those boxes or above each of those boxes. And then finally, we're going to do that same thing down the side. So hopefully you've been filling out your 16 box Punnett square as well. Oops. It's really easy to screw this part up too. So let's see here, as I did before. So you got to be really careful. All right, so. Okay, now here comes the part where we basically just drop and pull over. And this time you do have to try to get, for instance, all the alleles that go with the same trait together. And you do want to try to make it much easier to read when you're trying to extract the information from this Punnett square. You do want to try to write the dominant allele first, followed by the recessive. So you're just pulling down and then pulling over. So we've got 2 capital G's and 2 big T's. So just take some time and fill this out. Here's big G, capital T, little t. So I'll give you a moment to just fill all this out. All right, so this is hopefully what yours looks like as well, if after you've filled it all out. A couple of different things here. All of these obviously are going to represent the possibility or the probability that that offspring. So for instance, if we're just looking at one genotype, that would be 1/16 of all the possible outspring combinations, okay, for our offspring combinations for this F1 offspring generation would have green seeds that are smooth in color, okay. Down here, if you look, this is our only homozygous recessive individual or possibility, and that's 1/16 of all of these overall offspring. So this offspring possibility is going to be obviously wrinkly seeds that are yellow. Okay, now a lot of times people don't really care so much about what the genotypic ratio is of all of this. I mean, you can do the math yourself, and you're going to have 16, you know, hopefully take care of 16 boxes here and look through it. But the phenotypic ratio should be always the same when you're doing a heterozygous dihybrid cross with 2 heterozygous individuals. So I'm just going to shrink this a little bit and move it over. But if you're still looking at this, okay, what is the phenotypic ratio? Well, I'm going to show you a really simple way to figure this out. And it should be kind of easy, but basically you're just kind of connecting the boxes. So to figure out the most dominant, because we always start with the most dominant, go to the most recessive, you're going to make basically a connection of boxes. Now, wherever that line crossed through, that individual should be dominant in some way, either heterozygous or homozygous, okay, but still express the dominant trait for both genes, okay? So just double check. Are all these individuals green with smooth seeds? They should be. Yep, they are. This one does not count because the line did not go entirely through the box, okay? So if you count, there should be 9 individuals that are the most dominant, showing both dominant traits for both seed color and seed texture. So now let's work our way down. What's the next most dominant? Well, we've got a dominant here. And let's look for similar types of phenotypes. So we've got basically green seeds, but wrinkly seeds. So that's what we're going to look for next. Here's one with wrinkly seeds. So that's 2 that we've got total so far. And here's another one down here. Okay, wrinkly seeds, but with still a green seed color. You're always going to have a 9-3, 2. What's our next one? Well, here's another one. This one is yellow seed color, but it's smooth. Here's another one, yellow seed color, smooth, and here's another one. This one, I like to just make a little triangle, a small triangle, okay? So that's our next 3. So 9-3, and finally we're kind of looking for that last, it's usually the smallest number of the whole Punnett square. But we're looking for that last individual that is the most recessive, and it's going to be our homozygous recessive individual right there, and there's only 1 of them, and they're the last ones left. Now, because there are 16 boxes, if you add all this up, it should equal 16. So 9, 10, 11, 12, 13, 14, 15, and finally 16. So in case you didn't do it right, then you might not get that same number. So it's a good way to just check if you've done it correctly. So now, if you have probability questions, what is the probability that this F1 offspring of this heterozygous dihybrid cross will be homozygous recessive? Well, 1/16. Okay, and you can obviously make that into a percentage. What is the probability that these offspring will be homozygous dominant? Well, 9/16. Okay. Hope this helps. Again, I hope this helps, and we'll do more with the different, with Punnett squares with the different types of dominance in class. Thank you. Bye.